Think about the laboratory processes like the extraction, Taq polymerase, PCR optimization, primer selection, false negative and positives are important. Technical and biological replicates means a lot for your data set. Species specific or universal progresses that you choose are important. And the reference database, another thing that you must consider. For example, choosing the best barcodes is not always choosing is the optimum idea, because the barcode may be, or the gene region may be good to amplify, but in terms of if it lacks the reference database, then your data will not be annotated, or a taxonomic assignment will not be available, so it will be hard. So there's also another question, but I will answer them at the end of the lecture. So we can move on with the last part, the bioinformatics part, and then I will answer all the questions because the next lecture is canceled, so we have some free time. So the bioinformatics workflow is another important part. The bioinformatic processing of sequence information obtained after sequencing is one of the most critical steps of eDNA metabarcoding studies. This step helps to understand the research findings and components of the field and laboratory studies more clearly. Standardization of a set of codes called the pipelines in bioinformatics written on the terminals of computers ensures the quality and reproducibility of the findings. Some customization of this workflow is required for each study on its own. Therefore, converting the sequences into a taxon list after the raw read data is obtained requires multiple quality control steps. Some of these quality control steps are mandatory, while others are optional. We will discuss through them. There is no need for an absolute consensus for the approaches and the softwares used, because the software area is always open to change. There's always a better software or a better code that can be used with your operating system, with your desktop preferences. You might choose to work on terminal or graphical user interface. It all onto you. Some softwares are easy to use even with personal computers. Some of them are impossible to run in personal computers, but you need to run them in big servers. So before starting further analysis, it is recommended that the pretreatment steps that I will mention at the end should be carefully considered, at least for the high throughput sequencing data. But first, let's start with some terminology again, because these are the most common terminology in terms of bioinformatics. So the chimeras are the artifacts made of 2 or more combined sequences during the extension step of the PCR amplification. So because you have lots of different products, PCR products belonging to different species, sometimes they are accidentally, the polymerase starts to beginning part 5' side of one sequence and connects it with 3' sequence of another. So it becomes a chimera after that point. I will show how to remove them and how to understand if something is a chimera or a hybrid sequence. Another most common word or term in terms of bioinformatics is the Phred score. So the quality scoring for Illumina sequencing, providing the probability that a base call is incorrect. So as you can see here, there is the base call accuracy rates from 90% to 99.999% of correction in terms of Phred quality scores. So in terms of probability of incorrect base calls, so if your data has only one incorrect base calls in 1,000 base, then your Phred score is about 30 and your base call accuracy is about 99.9. I will give the thresholds for metabarcoding studies, which you should accept, which you should avoid. It's also all dependent to the study. Another thing is sequence merging. Sequence merging is the combining forward and reverse reads from paired and sequencing results using the criteria such as minimum overlap or the quality score. So by the way, I must say that another thing about the forwards and reverse reads in terms of high throughput sequencing, in Sanger sequencing, you can realize it from the letters F and R for forward and reverse. In high throughput sequencing, it's usually called R1 for forward read and R2 for the reverse read. So when merging your sequences, keep in mind that you should merge R1 and R2 for paired and sequencing. Another thing is the sequence trimming. You already know it from the Sanger sequencing, I presume. The process of cutting or removing the beginning or end of sequencing reads can be performed either by searching for a specific sequence, like the removal of adapters, indexes, and primers, or based on quality score. So some softwares will allow you to trim your sequences depending on their quality scores, remove the low quality sequences, or you can enter a sequence of adapter, index, or primers so that software can remove those sequences. You can also do it very easily, not practical, but just to give a information about the ones that never used it. You can put your sequence in a Word file, and then you can use Control F to find the primer sequence. And then when you find it, you can delete it from the sequence itself. It's that easy. The logic is like that. But of course, in a FASTQ file produced from high throughput sequencing, there will be not 1 or 2 lines, but instead millions of reads. So it will be hard to remove all of them. Also, you must consider while removing them, there's going to be mismatches. Like you have to put some threshold on mismatches. Usually it's up to 3 mismatches are acceptable for trimming the adapters, indexes, and primers, so that you should only look for the also different combinations with 3 mismatch threshold in your primers, indexes, and adapters. Another thing for another term is the singletons. Singletons at the OTUs that appear only once in the data are likely to be a rare taxa. They can be also false positives, or they can be low-level contamination, or unremoved chimeras, and should be treated with appropriate consideration. So they are called the singletons. You can see that there are different types of PCR errors. The red ones are derivatives of the real sequence and can be accepted. But as you see in blue here, the singletons appear only once in the data, and in the concept of high throughput sequencing, only 1 copy of a DNA is very unlikely to occur. So it's probably a singleton that should be removed. Other thing is the primer adapter trimming. These are not the terms. So let's move on to treatment steps that I mentioned before the terminology. So these are the treatment steps, and the first step is the primer adapter trimming. Preliminary steps of bioinformatics processing include the demultiplexing of the samples based on the indices used, the unique nucleotide tags incorporated into raw sequence data, and trimming, like the removal of the adapter sequences. The adapters are specific DNA fragments that are added during library preparation for ligation of the DNA strands to the flow cell during the high throughput sequencing. Additionally, the index sequences themselves and the primer sequences should be trimmed using the softwares. There are lots of common softwares like Cutadapt, Trim-O-Matic, KEIM, or NGS Filter of the OBI tools. They allow you either zero or a low level of mismatch between the exact sequence of the primer or index and the observed reads. So the mismatch level is up to you. And as I said before, up to 3 mismatches for the primers are acceptable in most of the metabarcoding studies. Merging or end trimming is the next step in your bioinformatics pipeline. Sequences from Illumina runs tend to drop in quality towards the 3' end of the reads, as phasing leads to increased noise and lower signal in later chemistry cycles. And it's for the 5' end in terms of the reverse reads. Thus, the quality score of reads should be reviewed to allow informed decision on the appropriate length of the trimming in single end runs, and merging for the paired end runs and subsequent sequence quality filters. Visualizing the quality scores from raw reads or demultiplex sequences using the software such as FastQC will help with the selection of downstream quality cutoff levels. When paired end sequencing is used for an amplicon of suitable size, the forward and reverse reads should be combined or merged to form the complete amplicon. Using merge sequence improves accuracy, as the lower quality bases at the tail ends of the individual reads can be corrected based on the combined reads. You can see it here. The minimum overlap for forward and reverse reads should be specified, and the remaining orphan reads with little or no overlap between forward and reverse pairs can be discarded. Also, overlapping is not an issue in some of the studies, but most of the studies is requesting overlapping. Inspection of the quality scores, as mentioned above, can provide an estimate of optimal parameters for merging of forward and reverse reads. Even though a specific consensus doesn't exist yet, in many cases, an overlap of at least 20 or more than 20 base pairs is selected. Quality filtering is the other step. For most high throughput sequencing platforms, a Phred score is calculated and subsequently used to determine the maximum error probabilities. Selected strategies include filtering based on a lower Phred score cutoff, usually set as least above 20 or 30, depending on your study. Quality filtering can also be performed based on maximum error. It's called max-EE probability, which is also derived from Phred scores. The lower the maximum error, the stricter the cutoff value will be. So selection of a maximum error filtering level of 1 or 0.5 is common in macroorganism studies. Additionally, in the case of single-end sequencing, or when long applicants without sufficient overlap of the forward and reverse reads are used, like in Nanopore or PacBio sequencing, it is advised that trimming should be performed from the appropriate end. It is often the case that reads are trimmed to a common length, which facilitates alignment downstreams and minimizes miscalled bases as a merging step that cannot be used. Another issue is removing the short reads. Many studies also select to remove the short reads from the data set before clustering, as the presence of high length variation. It could influence the clustering processes. These sequences could result from sequencing of primer dimers that have not been removed. Different studies select a variety of minimum length reads from very short 20 base pairs to medium, like 60 to 80 base pairs, and up to 100 base pairs. Some demultiplexing or quality filtering workflows may automatically set minimum sequence length when processing input data, and it's advisable to check whether such a parameter is included by default. So in terms of Illumina sequencing, the expected view of the FastQC analysis should be like the one on the right, because depending on the chemistry you are using on the Illumina high throughput sequencing, there can be only one outcome in terms of the size. So there is no length difference between the sequences in Illumina sequencing. But in other platforms like IonTorrent or Nanopore sequencing, the lengths can vary like this. And if you are using, for example, a 100 base pair long chemistry in Illumina sequencing, so the graph should look like this. Removing singletons and chimeras is one of the most important steps after OTU clustering. The chimeras are the byproducts of PCR amplification process from 2 or more parental sequences. That's why they are called chimeric. So most commonly produced through an incomplete extension step, like the tag jumps and the index jumps. It has been shown that when unique reads such as chimeras or singletons are withheld in analysis, the estimation of diversity can be severely inflated. The nature of the chimeric sequences, which can be present as high quality reads, doesn't enable their removal directly through quality-based trimming. Removal of chimeras can be performed either de novo or based on a reference database. The most common practice to date is the de novo method, as a sufficient reference database may not be available at all. Despite the variation in softwares used such as UKIME, ObiTools, it's the ObiClean command, I guess, and the chimeras layer, there is a consensus regarding the importance of removing chimeras and singletons as a minimum quality control for bioinformatics pipeline. So the minimum information requested from your bioinformatics filtering will be at least you should remove your singletons and chimeras from your dataset in order to make it a publishable dataset. Another issue is the abundance filtering, which is very important in terms of metabarcoding. Also, if you are looking for relative abundances. In addition to quality filtering based on quality scores and removal of chimeras and singletons, many studies also employ further filtering for removal of low abundance sequences. This step arises from the need to control for laboratory contamination or because of cluster contamination on the flow cell. It's unique to Illumina platforms. The process of applying abundance filtering requires setting an OTU abundance threshold by which OTUs are only retained in analysis if their relative abundance is higher than the selected threshold. Selection of a threshold varies between studies, and there is no generally accepted definition of what constitutes an insufficiently abundant read, perhaps with the exception of the singletons. Abundance filtering may be applied minimally, or it can be avoided entirely, especially if stringed quality trimming parameters are applied to raw reads and detection of rare OTUs is an important aspect of a study. Another option that could be used involves selection of a threshold based on availability of empirical data as it was performed in a study. An increasing number of studies have employed the sequencing of positive controls to establish a threshold level. Technical replicates can also be used to assess the consistency, as was shown to be effective with assessing the diets of species. Using a positive control defined error level works by identifying the abundance of sequences in the control sample that belong to non-target taxa and can be the result of errors such as contamination. Furthermore, the distribution of fixed reads assigned to target samples has been used to investigate the presence of tag jumps and misassigned reads during demultiplexing. The exact mechanism for misassignment of reads remain unknown, but increasingly many studies are reporting this error to be between 0.01 to 0.03% of reads. Adjustments for this include use of a threshold approach based on negative and positive controls and remove a low number of reads from any given sample. The issue of abundance filtering most significantly causes uncertainty in low abundance OTUs and will continue to be a problem for the detection of rare species. Therefore, to avoid negative impacts to scientific insights or management decisions, careful consideration and transparency regarding how technical artifacts are dealt with during bioinformatic data analysis is needed until these artifacts are well understood. So in terms of the transparency of the data, recording to remote data is of a great importance. For all quality control steps, the data removal should be transparent. Often studies report the total number of sequences obtained, but then rarely show how each quality filtering step affects the number of sequences used in testing ecological hypotheses, nor do the researchers provide the subset of sequences that were retained or omitted. When you modify and deleting data without a clear justification, it doesn't allow transparency in terms of peer review, in terms of reproducibility. Therefore, including a supplemental table in metabarcoding studies showing the number of sequences remaining after each filtration step is advised. And archiving the subset of reads retained after each filtering step on a platform such as Dryad, or archiving the exact pipeline with version control information on a platform such as GitHub will allow for greater transparency and reproducibility of quality filtering. In order to give an example pipeline, in my lab, we usually use the ObiTools pipeline. We modify it according to the rest of the parameters in the study, but it has some different packages works inside the pipeline. So first you put the raw reads forward and reverse reads into the pipeline. The first, and if your run is paired-end, you use the Illumina paired-end command for the microassembly of paired-end reads to have consensus reads of merger reads. And then you use the NGS filter, which needs a sample description CSV file. In this file, you write the names of the tags, the adapters, the primers, name, or the information about the samples that you pulled together. With this NGS filter code and with this file giving all the information, you can demultiplex all your samples and you can assign all the reads to separate samples in that step. And then there is a tool called ObiAnnotate. It will add the sequence length as the new attribute. Here, ObiGrep is working like the Linux command grep. All it's do is to filter the erroneous sequences. You design the parameters yourself, like the minimum length should be 100 base pairs, the maximum length should be 200 base pairs long. And also you can include number of counts in your data set. So you can say that only keep the sequences that exist more than 5 times or 10 times in your data set. We will make it in the other OBIGREP, but it is what OBIGREP does. You give the parameters and it filters according to that parameter. And after that, we use the OBIUnique package, which is the dereplication of the sequences. So you have millions of reads, which can be clustered into replicated sequences. So what it does is takes those replications and cluster them as single clusters and keep the records for how many reads it have. Again, in here, with the OBIGREP, you can filter the singleton sequences from your entire data set. And then we use the OBICLEAN, which, as I mentioned before, filters the putative PCR and sequencing errors with the threshold that you determine, like 1.0 or 0.5 will be enough for a metabarcoding study with a Phred score of 30. And then the final OBIGREP step, in which you filter the sequencing errors that were cleaned at the OBICLEAN. And then the ECHOTAG step will do the taxonomic assignation. In order to do that, ECHOTAG needs reference sequences, as you have with your local database constructed, build it using Sanger sequences. Or you can take NCBI, EMBL, or SILVA databases, turn them into OBI taxonomy format, and using those taxonomy information, you can feed your ECHOTAG step that I need to assign my sequences with the taxonomic database. So it will give you the sequences. There is a sub stage called the T for the annotated sequences to be diversed into FASTA files. You can use it. But finally, at the end, you can use the OBITab to convert them into tables like the spreadsheets or CSV files with annotated sequences. Your sequence, number of reads, number of reads, the sequence name, and how many, and the grouping according to your multiplexing selections. So, what to consider from the bioinformatics part? It's the quality check is the most important part of it. Trimming, filtering, abundance filtering, short reads filtering is the main filtering steps that you need to apply to your datas. And it's in terms of case specific pipelines, it is important to design your own readings according to them. I think we have some time for give some examples because our next lecture is canceled. So I will just give 1 or 2 examples from our studies. Let's start with the bioinformatics one, like the mock DNA study. This is results from a mock study. What we do is approximately we extracted DNA from tissue samples of 600 freshwater, marine and brackish water fish species from European freshwater and marine and brackish waters. So it's a DNA cocktail containing 600 different fish species DNA. We designed to put same amount of DNA into that cocktail. So concentrations are all the same. We mostly use 12S primers, the MyFish, Teleo, and AMPD primers to check the bias of the experiment. So normally what you expect is you have to see the 600 species like very similar abundance levels. But when we check the data set, the variation about the abundance levels, the relative abundance of the species, even they are included in same concentration levels and all extracted from tissues, the variation is huge, like 0.001 to 8.7. So some species exist in like the 10% of the experiment. So this variation is seen in qPCR, digital PCR, and also a high throughput sequencing in terms of copy numbers, and in qPCR and digital PCR in terms of DNA numbers. So the thing here is the bias, but what bias? It's the primer bias. So the primer, the universal primers that you use have a bias towards the ones that it fits better. So when we check here, this Capoeira sample, or this the second one or the third one or the top 10 ones are more compatible with your universal primers. So if your primers are easier to go and attach to them, then their relative abundance will be large. So you need to optimize a data like this regarding, you should consider the primer bias, and according to primer bias, you need to optimize these values and normalize them according to the fact that they should be all at the same level. So when you make a real-time, real-life sampling from European freshwater or European fish eDNA monitoring study, you should realize that 8.7 in terms of Capoeira means 0.001 in terms of another species. So there is no such primer that has no bias towards any of the species. Another bias regarding to local and open database is in terms of our own local database, there are 34% different species profile in the entire data set. So the results obtained using our local reference database and the results obtained using our using EMBL database gives 34% different species structure. Another thing is in the EMBL database, 41% of the species never exist, which is something well expected as the Mediterranean region is full of endemic species and they did not exist in the EMBL database. At least the 12S primers or the region that we amplify doesn't exist in the database. Another thing is the pipeline comparison. With the different parameters that you used in your pipeline, you can enhance your result 14%. The results can be gathered with a modified pipeline. So just with playing with the things like the trimming, filtering, abundance, short reads, or error levels, or quality control, you can enhance your data to give you 14% more species with a modified database. So here are my concluding remarks. So I can say that in the near future, the capacity of analysis performed from eDNA samples obtained from communities of target organisms to provide information on all biodiversity will increase with the rapid development of the most appropriate field, laboratory, and bioinformatic tools required for the eDNA metabarcoding technique. In order to simultaneously increase the sensitivity, accuracy, and ecological and social relevance of environmental DNA, we need to better understand the spatial and temporal relationship of eDNA and species. Errors and uncertainties associated with existing eDNA metabarcoding studies can often be overcome by carefully designed studies, appropriate primer selection, and correct sampling methods, easy as that. Over time, the scientific cycle that developed eDNA metabarcoding methods will reduce the uncertainty in the analysis of positive and negative eDNA findings for target species, creating motivation for the development and wider use of ongoing eDNA metabarcoding methods, thus enabling eDNA metabarcoding to predict species diversity. It will result in it being considered a method comparable to other methods. Metabarcoding of the eDNAs of species in the coming years will both be complementary methods to currently used methods and will accelerate our understanding of biodiversity in our planet as a standard research tool. In this seminar, I gathered the results and knowledge of leading international scientists and research groups in order to be able to apply eDNA metabarcoding methods and to lay the foundation for a concrete framework of existing practices. The recommendations set out in this seminar are a basic guide for applying metabarcoding methods, considering the limited time. However, each study is different and the exact protocols to be used vary according to the field of the study and the questions and the hypotheses addressed. So the eDNA-based techniques will also change and advance with the development of technology. For those who who interest in bioinformatic pipelines of how to filter and trim the raw data gathered from high throughput sequencing, I will be happy to help you. My email address is in the video. You can check it later. So if you want to get a detailed information or even an online practice on how to analyze this data, please contact me. And if you have any questions, you can ask it or send it to my email. So I would like to say thanks, teşekkürler, and efaristo for all the participants of this seminar. If you have any questions, you can write it through the chat or send me an email. I don't want to waste your lunchtime, and you have a long lunchtime regarding the cancellation of the next lecture. So I can answer your questions, or you can write them for me for a later conversation. Thank you.